This is Laith and Jordan here. Our article was a cell cycle automaton model for probing circadian patterns of anti-cancer drug delivery. So first off, we're going to start with some biological background. So first off, just starting like super simple and basic, what exactly is an automaton model? Essentially what it is, it's simply a phenomenological description of the cell cycle. It allows for sequential transitions between the phases around a mean duration, mean basically like the time it takes to transition from one phase to the next. To prevent the model from running to infinity, basically continued cell growth, they induce a probability of cell death. And our model in this case revolves around a 22-hour cell cycle, and this 22-hour cell cycle is based off previous cell distribution experiments that were conducted. And you can see below, the M phase is 1 hour, G1 is 9, the S phase is 11 hours, and then the G2 is 1 hour for the total of 22 hours. So biological background continued. So now we're going to talk about circadian rhythms. Just a sort of simple explanation. According to the National Institute of General Medical Sciences, it's defined as the physical, mental, and behavioral changes that follow a roughly 24-hour cycle responding primarily to light and darkness in an organism's environment. They are found in most living things, including animals, plants, and many tiny microbes. The study of circadian rhythms is called chronobiology. And you can see in the figure to the right here, this is sort of a simplistic figure that sort of describes what a circadian cycle is and what's happening to your body. So in this case, 6 a.m. is morning and 6 p.m. is dusk. So you can see at noon, you're most active, you have a high alertness, best coordination, and as you circle around, your body sort of changes. So you have different reaction times, different cardiovascular efficiency, blood pressure, melatonin, bowel movements become suppressed. And then at 2 a.m., you're in your deepest sleep. And then again, as it goes around 6 a.m., you're now awake, sharp rise in blood pressure. So this is sort of the generic circadian pattern that humans see. So again, a little more on circadian rhythms. They are self-sustaining, meaning that they adapt to the environmental changes via the internal cues of circadian genes and proteins that originate from a pacemaker. AKA, this is the circadian nucleus, and it's located within our hypothalamus. Now, the interesting thing about this is that there's actually 10 to 20,000 neurons located within the SCN, the circadian nucleus, and each neuron is actually capable of independently generating self-sustained circadian rhythms. So it's a lot of rhythms. A lot of them are yet to be explored by scientists. Entrainment, as our article describes, is the adjustment of these rhythms. Our article mostly focuses on the light and dark transitions. So entrainment can occur based on time zones, temperature, and light regulation. So for example, jet lag, when you transition from a new time zone and you experience tiredness, drowsiness, and sometimes you even just feel sick, it's your body's way adapting to the new cycles now. So with a new time zone, you have new light and dark phases, and it's just your body's way of compensating for that over time. And you can see in the figure on your right, you see the light input into this circadian nucleus. Your output is the rhythms, physiology, your behaviorals, your body changes, and everything.
So 5-FU, let's give you some biological background of what we're talking about here. 5-FU, which is otherwise known as fluorouracil, it's an anti-cancer drug that works through the irreversible inhibition of thymidylate synthase. Now this inhibition, it'll disrupt the synthesis of thymidine, which is an essential deoxynucleoside for DNA replication. But because of the irreversible inhibition, 5-FU and most other cancer treatments will necessarily have harmful effects on both normal and cancerous cells. And what you see here in the reaction down there is that you see the 5-FU, which is accepted by thymidylate synthase as a substrate, and that'll react, as you can see, with thymidylate synthase and methylene THF. The three of those will be joined in a covalent intermediate, and at this stage, the fluorine in position 5 of the uracil, as you can see, will resist the abstraction by the base of the enzyme. Now, the covalent intermediate will be locked up because the enzyme remains irreversibly locked up at that stage because of the fluorine in position 5. Inhibition of thymidylate synthase will necessarily starve the cell of thymine, which is required, again, for DNA synthesis. And as such, DNA cannot be synthesized, new cells cannot be born, and as such, you have a lot of other harmful effects as well, such as hair loss, such as your entire immune system. Everything, you get tired. So all the negative side effects of anti-cancer medications are because of this right here, of the synthesis of thymidine right there. Now, because, as you mentioned, all cells are affected by anti-cancer medications, the study of chronobiology is very important to help mitigate healthy cell loss and maximize the efficiency of these anti-cancer medications, such as 5-FU. Now, because 5-FU is most toxic to cells that are undergoing DNA synthesis, the specific cellular metabolism rates at different times of the day plays a major role in the tolerability of 5-FU. So after a lot of independent research, sinusoidal modulation of 5-FU between 10 p.m. and 10 a.m. was found to be the optimal time to reduce toxic effects of the drug. As you can see in that graph right there on the bottom, you have a sinusoidal modulation of 5-FU, and that comes up only at the lower points of DNA synthesis of cells in that phase. And it was found that the optimal time was at 4 a.m. Doing so at 4 a.m. and introducing 5-FU at 4 a.m. gave patients a 40% increase in tolerability of doses, which means that you can maximize the benefits of 5-FU and minimize the healthy cell loss by doing so. So circadian rhythms have been researched for quite some time. They were first discovered in mammals in 1994 with mice experiments. The first discovery of the clock gene, and it's called the clock gene not because it's about the circadian clock, but it's actually a clever acronym, the circadian locomotor output cycles kaput. That was discovered in 1998. What was interesting in that same study is that mutations of this clock gene were found to be heritable, but also reversible. So they had mice that they bred to have longer daily activity, up to a 27.5-hour circadian rhythm. And then as opposed to control mice, which for some reason have less than a 24-hour on average circadian rhythm at 23.3 hours. They also subjected mice to jet lag, and they saw that it naturally resets after 8 whole days of being involved in the new time zone and the new light cycles. Light patterns in those studies and subsequent studies revealed that the clock BMLA1 protein complex that regulates the period of WEE1 expression. And now it's important because WEE1 inhibits CDK1, and CDK1, as we know, triggers the G2M transition. So the inhibition of CDK1 will therefore inhibit the G2M transition, and that's all done with induction of clock BMLA1. And therefore, the clock BMLA1 provides a direct mechanism that connects the cell cycle on a daily basis to their circadian rhythm.
So for our model, it's actually split into 2 different components. We have the WEE1 and CDK1, which sort of lays the foundation for our 5-FU model. And as Laith had mentioned when he was discussing WEE1 and CDK1, as you see in the figure bottom to the right, CDK1 acts as the activator for the G2M transition, where WEE1 is the inhibitor, and this is all based off the circadian clock, which is our light and dark transitions. So starting with our WEE1 CDK1 model, you have at the top the survival and death rates. Then as you continue down, you have the amount of cells where each phase, where tau is the duration of each phase, the time it takes to transition to each phase. And then you have down even further the probabilities of cells not dying in a given phase. And then over to the right, probability of phase transition based off WEE1 and CDK1 rates, and the rates being Kc and Kw, as you see for CDK1 and WEE1. And then lastly, you have the cell duration with variability. And variability in this case is just saying when the phase of a cell cycle occurs. So they're adjusting those times that we described earlier, the 11 hours, the 9 hours. The variability induces a variation in those times, so they can actually occur prematurely. So now let's discuss some of the MATLAB code for our WEE1 and CDK1 model. Now this MATLAB code here that you see is not actually done by us. So this was actually provided from a reference standpoint for the WEE1 and CDK1 model, and this reference will be added to the file presentation folder on ELMS for you guys. So this was done by Altanach and Gans, and it basically just is the code for all the equations described earlier. So you have your durations of the cells, the variability that they induce, the probability of cell death at any time in the cell cycle, logistics, which is sort of their random logistic, basically saying like when and when not to induce the probability of death, because cell death can occur at any time. It's not just a simple rate constant that you can predict. Cells live and die at any given point in our body. Then you have the entrainment of the circadian clock. Again, that's set at a random point, so it's all distributed randomly. And then you have the amplitude of WEE1 from 0 to 100, CDK1 0 to 100, how long the duration lasts, which is 12 hours, and then the relative effect of each one as well, like how much their rate is affecting the overall system. So here you have the WEE1 and CDK1, our results. So let me mention that we're going to be adding the actual MATLAB code in its entirety to ELMS for everyone. The code itself, we gave a little snapshot of it earlier. The actual entire code is 374 lines long. So it's an exorbitant amount of code. It's a pretty substantial file. Again, this was years worth of research that they put in. So our job with this code was to sort of dissect it and figure out how they used this to achieve their results. So by doing so, we were able to play around with some of the variables and sort of replicate what they got in their references. Again, this WEE1 and CDK1 model lays the exact foundation for our 5FU model. So that's why we're sort of discussing it and what they did. So starting at the top, you have 0% variability. And again, we were able to replicate this and show on the graph on the left. Then you have 10% variability and 50% variability. So what this is telling you is that at 0% variability, the cells are completely in sync with one another. The transitions that we described earlier are exactly the same. So at 11 hours, at the end of that, it automatically transitions to the next phase, and then transition to the next phase. They're perfectly in sync with one another. So at 10% variability, the cells are becoming less synced with one another. That's why they're not a square. They're not square wave anymore. They're more rounded. So at 50% variability, what this is saying is that essentially after your first initial cell cycle, all the cells will become completely desynchronized. So the reason that it looks more like a straight line is that all the cells and the cell cycles are reaching towards their steady state. So the lower the variability, the more your cell cycle are completely in sync with one another. So you get the transitions are exactly one after the other. As your variability increases, the cells become less synced, and they'll reach towards steady state quicker. So again, showing the WEE1 and CDK1, our results. Here we changed a number of variables to match their figure. We changed the variability at 20%. An initial number of cells is 10,000. The probability is 0.000525. And the logistic regulation, aka the rate of death, is 0.001. So you can see we were able to match their figure. And the reason this figure is so important, as you can see on the left, as we described, WEE1 is a inhibitor to the G2M transition. So as you can see, if you look closely, a rise in the Wee1 dips the number of cells downward in the M phase transition. So you can see with a rise in Wee1, the red line there for the M phase goes to 0, meaning there are no cells in that transition because of Wee1. And then as Wee1 decreases, you see a rise in CDK1, thus allowing the transitions to occur again. And this all occurs from Wee1 and CDK1 based off the circadian pattern of the light and dark transitions. So the line at the very bottom of the figure on the left, the white is light and the black is dark. And this will become a key component for our 5FU model, which we will describe.
So our model in our paper is a 5FU model. So right off the bat, you might be wondering why we went into such detail about the Wee1 and CDK model previously. But the reason for that is because the authors of our paper, they simply added 5FU just to make sure that this 4 a.m. time is best based on the Wee1 and CDK model. And as you remember, Wee1 and CDK influences the G2 M phase. And so essentially what these authors did was prove that 5FU was the most beneficial at night between 10 p.m. and 10 a.m. with a peak at 4 a.m. So the variability is the same as the last model, as the previous model. As is a lot of their graphs. And so what they did over here, if you can see, these graphs are from this author's report, and they only looked at cells in the S phase. And that's an important distinction, which is why these graphs look a little bit more simplistic than the others. If you take only the S phase from the previous graphs as well, you'd see that they match up as well. And here we're able to replicate the data and replicate the graphs that the authors made using 5% variability as well as 15% variability. And again, remember that the peaks are different here for each time because the authors do induce a bit of a probability factor into this as well, just to make it more natural. And if you see here, everything mirrors to what the authors were doing. These next slides are also, we were able to replicate their graphs as well as their data. Over here, what you're looking at is our MATLAB code, which we generated. The top two, they don't exactly match up, and that's mainly because, again, the authors here were only looking at the duration between 10 p.m. and 10 a.m., and so there was a lag phase before 10 p.m. and after 10 a.m. So we were able to replicate their data between 10 and 10, then at night. The bottom two graphs are the probability to quit the cycle, and that's the cell death. And so over there you can see a near exact match to what they introduced in their data.
All right, so let's bring this whole thing home now, bring this all to fruition, and sort of show you how the Wee1 and CDK1 and 5FU compare to one another. So again, the reason we went through all the explanation of the Wee1 and CDK1 is to show that our authors simply used all the previous studies to show that 5FU, based on circadian cycles in the light and dark transitions, at 4 a.m. will best reduce the cytotoxic effects. So as shown in the graph below, the article directly relates the two, as I said. So you can see in the graph to the right, you have 5FU peaking at 4 a.m. for the S phase. And then as shown in the arrow to the left, this is from our reference article. You can see that the S phase is almost a mirror image of one another. And then something else to note is that the CDK1, shown below, during the light and dark transitions, directly mirrors 5FU. So essentially what the authors did is that they represented 5FU similar to CDK1, but instead use CDK1, in this case 5FU, as an inhibitor to the S phase. So that kind of brings everything all together, puts the whole thing into perspective as to all the previous studies and the WEE1 CDK1 model and why their code was so important into the discovery of the 5FU model.
So what is the goal here of these studies? So the main purpose is to reduce the cytotoxic effects of anti-cancer medication with entrainment to circadian rhythm, synchronization. So by directly using the same modeling criteria as the WEE1 and CDK models, these authors were able to prove the validity of previous clinical trials of 5FU into a model. And so this model, unlike the WEE1 and CDK models, was able to probe and search for various times throughout the day in order to determine when a specific cell phase was occurring. That's important to be able to minimize to figure out when cells are less likely to be in the S phase to introduce anti-cancer medication. So in doing so, the model is able to predict the likelihood of when cell activity is at its lowest, and it does that by measuring the output of the number of cells in that phase based on the input of the anti-cancer drug, 5FU, all relating it back to time. And so ultimately, this really does improve on targeting specific phases, like the S phase, during their least active times for infusion of anti-cancer medication, which again increases the tolerability of patients to 5FU.
So again, what is the goal? As Laith mentioned, the main purpose is to reduce the cytotoxic effects of anti-cancer medication with the circadian rhythm, and this is synchronization. So we can't state it enough. This is the biggest takeaway here. So as shown in the graphs below from our article, the figure A on the left is the absence of entrainment to circadian clock, and this is with a 4 a.m. 5FU delivery. So as you can see, with any variability change, you have an exponential increase in the cumulative number of cell deaths with any variability with a 4 a.m. delivery, which is supposed to be the best. Now, the reason is because when you're not entrained to the circadian clock, the cell cycle is simply just in a sinusoidal manner. It just goes up and down without any lag phase. So it's not taking into account light and dark. So when you deliver the drug at nighttime, the cell cycle doesn't recognize that. It just simply takes in the 5FU drug during its sinusoidal manner. And so that's why you see, because it's not being controlled by light and dark, you see that rise in the cumulative number of cell deaths until it eventually reaches steady state. There's nothing controlling the cell cycle. Whereas in figure B, you see that with the presence of entrainment circadian clock with the 4 a.m. delivery, this dramatically decreases the number of cells that die. Now, the reason this is because, as I mentioned, that when you entrain the cell cycle to the circadian clock, it's now synced with light and dark. So if you think about it, when you bring in 5FU during the dark phase, and this is during your least active time of the S phase, this mitigates the effect of the 5FU. You're not going to get the cell death as you would. So that's why I see with any variability increase, you will decrease the number of cells that are killed because you're controlling your cell cycle by the light and dark transitions. And that's the main takeaway here. So now the limitations to our model. So for our model, it doesn't actually take into account what is happening on a molecular level as the cell cycle is occurring. So meaning that there's no rate constants, there's no kinases. It's a simplistic phenomenological approach for our model. So it's not taking into account everything that's happening during a cell cycle. DNA replication, transition, nothing of that nature. It's simply just a simplistic approach to describing phases and how it relates to anti-cancer medication. In addition, the probability of cell death is at a constant rate in our model, unlike the WEE1 and CDK1 model, which introduces a randomized probability of that function. In addition, the circadian entrainment is based only on light and dark patterns. For example, a peak activity at 4 p.m. or 4 a.m. So as we mentioned earlier, for all the neurons that are within our hypothalamus, our body self-regulates numerous patterns of temperature, time changes, sleeping patterns, and everything in that nature. So scientists can't take into account every single environmental effect and all the neurons and all the changes that our body is doing without us even being fully aware of it. They're simply only taking the simplistic changes in light and dark and being able to relate that to the cell cycle. Okay? Now, food for thought, just sort of something to think about for the future, if this interests you. How would circadian rhythms change for people such as in like Alaska, where they could have a full 23-hour day of light, or they have 32 or 30 days of darkness, or people who are blind, or as we mentioned, traveling to different time zones for in case of light and dark patterns, as we mentioned, jet lag. So all of our rhythms and all of our changes occur for each individual person. It's all different. It's all based on where we're at, where we live, and how our environment changes us as human beings. So lastly, it also assumes that the 5FU is directly relatable to the WEE1 and CDK1. So as we shown earlier, CDK1 is directly related to 5FU. So they're just assuming that if they change that and introduce 5FU, that nothing else needs to be taken into account, that those two are directly related, and they just change CDK1 to 5FU and say, here's our model, it inhibits the S phase, and these are our results. So those are our limitations.
So why is this all so cool? So this model can be used for scheduling various types of anti-cancer medications that inhibit other phases of the cell cycle. Its versatility and reduced parameters will provide framework for future studies. This is so important to us as a populace because cancer chronotherapy is, one, currently happening in a lot of clinical trials like 5FU, but is also currently happening in general in cancer patients. Chemotherapy drugs are infused into patients either overnight, either via IV or time-release capsules, but also you can nowadays wear these CAD devices, CADD devices, which are special portable pumps essentially that deliver chemotherapy on a chronomodulated basis. A lot of them are small, battery operated, and portable as well. And so you're able to kind of sync up the models that we just spoke about and your actual treatments. Now, this can obviously be expanded to take into account molecular genetics, environmental changes, and like Jordan was saying in the beginning of this presentation, I mean, there's each one of the neurons have their own special circadian rhythm, and so there's still a lot more research to be done, but it seems like we're really on the right track here to provide the most drugs and to make that most tolerable for patients.
So now our question for you guys. So as we mentioned, variability as described in the model is the random transition to the next phase of the cell cycle. This in turn dictates their synchronization with one another. So for part A, if the variability continually increases, what will eventually happen to the cells? And here I give you the hint. If you refer to the page 1049 in our article and refer to the left column, the second paragraph, I guess, you should easily arrive at your answer. And part B, what happens to the cells when the variability is at 0%? So we actually just talked about this earlier. This should be earlier mentioned in our video. The figure you see below was actually shown already, and I had mentioned what exactly is occurring at 0%. So if you go back, take a look, and really think about what the figure is showing you, you should be able to arrive at your answer.
So thanks for tuning in, guys. We know this might be a bit difficult doing this online, but hope you enjoyed our presentation. If you have any questions at all, please don't hesitate to email us at laith.abutalib@gmail.com or jordan.hackert@gmail.com, and we'll be as responsive as we can. These are our references. The first one is the WEE1 CDK model, and the second one is, of course, the 5FU model that we focused our presentation on. Thanks again.